Let's talk about the cell cycle. Cell, as we already know, is the structural and a fundamental unit of life. Now, the cell undergoes a series of divisions and duplications. It is therefore the series of events that takes place in a cell leading to its division and duplication. This has been named as the cell cycle. Each cell is capable of division while passing through a cycle, and hence it is the cell cycle. There are three types of cell divisions, namely the amitosis, the mitosis and the meiosis. Here we can see the pictorial representation of a cell cycle. The interface has the G1 phase, which is the first growth phase, the growth and normal metabolic roles. Next is the S phase or the synthesis phase, wherein the DNA replication takes place. G2 phase is the growth and the preparation phase for mitosis. Then comes mitosis or the M phase, which has four divisions: prophase, metaphase, anaphase and telophase. What is amitosis? In amitosis, the chromosomes are not visible. The cell division takes place, simple cleavage of the nucleus without spindle formation, and therefore the chromosomes are not seen. It is found in simple forms of life such as the amoeba, bacteria. The nucleus elongates in a dumbbell shape, followed by the invagination of the plasmalema. The constriction in nucleus as well as the plasmalema deepens and divides one amoeba into two daughter amoebae. What happens in mitosis? The cell cycle consists of a long interphase followed by a short mitotic or M phase. The interphase is the phase between two successive cell divisions. Metabolically, it is an active phase. Interphase is the phase of the cell cycle in which the cell spends the majority of its time and performs the majority of its purposes, including preparation for the cell division. In preparation for cell division, it increases its size and makes a copy of its DNA. Interphase is also considered to be living phase of the cell, in which the cell obtains nutrients, grows, reads its DNA and conducts other normal cell functions. The majority of the eukaryotic cells spend most of their time in the interphase. It does not describe a cell that is merely resting, but is rather an active preparation for cell division. A common misconception is that interphase is the first stage of mitosis. However, since mitosis is the division of the nucleus, prophase is actually the first stage. Here we can see four phases of an eukaryotic cell cycle. We have already discussed that the interphase has the G1, the S and the G2 phases prior to the beginning of the mitosis. What happens in the G1 phase? The cell grows and functions normally. During this time, a lot of protein synthesis occurs and the cell grows to about double its original size. More organelles are produced, increasing the volume of the cytoplasm. If the cell is not to divide again, it will remain in this phase. It is also known as the post-mitotic phase. Next comes the synthesis phase, in which the cell duplicates its DNA via semi-conservative replication. The DNA molecule of each chromosome replicates by the synthesis of a new DNA molecule on the template of the existing DNA. The cell retains the original 2n number of chromosomes but now has duplicate sets of genes. Synthesis of histones and proteins also occurs. The cell has double the amount of DNA present in the original diploid cell. Next comes the G2 or the gap 2 phase, in which the cell resumes its growth in the preparation for mitosis. This is known as the pre-mitotic gap phase. In this phase, duplication of centriole, the mitochondria and the chloroplasts occur. The spindle proteins are also synthesized. Synthesis of RNA and proteins continue. The decision of the cell to divide occurs in the G1 phase. If the cell does not have to divide, then it enters the G0 phase or the quiescent phase. As soon as the conditions change, the cell enters back into the G1 phase. Therefore, the G1 phase in the cell cycle is also known as the restriction point or the checkpoint. As the cell crosses the restriction point, rest of the cell cycle is completed. The two factors which determine whether the cell has to divide or not are the surface area ratio is to the volume ratio. A cell should have high surface area and high volume area, and the karyoplasmic index. Question was asked in the 2005 AIPMT, at what stage of the cell cycle are histone proteins synthesized in an eukaryotic cell? And the questions were during G2 stage of prophase, S phase, entire prophase, or during the telophase. The S phase is the synthesis phase of the cell cycle. Hence, the correct answer is the S phase. Let's talk about some of the facts. G1 to S transition is carried out by G1 cyclin plus CDC2 kinase. G2 to M transition is triggered by the maturation promoting factor formed by the mitotic cyclin plus CDC2 kinase. Nucleus attains its maximum size. The cell cycle is regulated by cyclin-dependent protein kinase. What are cyclins? They are kind of proteins that activate protein kinase regulating the eukaryotic cell cycle. Onion root tips or other meristematic tissues are taken to study the process of mitosis. Mitogens are substances which induce mitosis. They are the auxins, the cytokinins, the gibberellins, and the insulin. There are certain substances which inhibit mitosis. They are azides, cyanides. Cyanides, they inhibit the process of prophase. Mustard gas agglutinates the chromosomes. Kilones are small peptides or glycoproteins in the extracellular fluid which inhibits mitosis. Colchicine is a mitotic poison inhibiting the formation of spindle by causing the disassembly of microtubules. G2 phase is followed by division involving nuclear division, that is karyokinesis, and cytoplasmic division, that is cytokinesis.